Okay, good morning to everyone. My name is Raquel del Campo. I'm a PhD candidate here in the iuFOR-UVa, and today I'm going to talk about the analysis of the virome of Phytophthora species isolated from alders. First, for those who don't know, I'm going to talk a bit of the disease. The alder decline is a widespread disease that affects all alder species, and basically is characterized by the progressive decay of the tree. That the main symptoms that you can see is these bleeding cankers in the bottom of the trunk. Then you see in the leaves chlorosis and also microphilia, the death of the apical branches, and also finally this disease ends with the death of the tree. The main cause that it's been identified is the Phytophthora. I'm not going to say a concrete species because we are not sure yet. In the bottom of the slide here, you can see all the Phytophthora that we've been able to isolate, affecting all those alders. And what I want to mention here is that Phytophthora are really not real fungi. They are omycetes. That is kind of different. They act like fungi, but they are not real fungi. The main difference is that they spread asexually by zoospores. That's why this disease is spreading really fast, because it's spreading through the waters. And the alders live in the riverbanks, so it's kind of like an important point to mention. And also different with these Phytophthora and the real fungi is that the walls are based on cellulose instead of chitin. That's an important difference. But in this presentation, I want you to be focused on the viruses. I guess that everyone knows that any organism can be infected with viruses. Even the most tiny bacteria, or even us, we can be infected by viruses. So do the fungi and the omycetes can be infected by viruses too. The difference mainly is the genome. They can be formed with DNA genomes or RNA genomes. And I'm going to be focused on mycoviruses and omycete viruses that mainly have RNA genomes. So I'm mentioning this because as all the viruses, all the mycoviruses can have effects. In this case, mycoviruses can have three different effects. They can modify the behavior of the host by making it less pathogen, so it would have a hypovirulence effect. They can have no effect, so they live together, no effect, they are cryptic, or they can be, they can have a potential in the pathogenicity of the fungi and making it more virulent, so they will have a hypovirulence effect. These effects are important when we are talking about biocontrol. In the case of the hypovirulence, we are focusing more on the pathogens from plants, making less pathogen so the plant can survive. And in hypervirulence, we are talking more about pathogens that will affect to the pest, so we will kill the insects more sooner. So basically, last year I was here talking about the theory, what I was going to do. I presented these objectives, making a lot of questions to myself and not having the answer to it. But this year I'm focused on the first objective of my thesis, because finally I achieved to study the whole virome of the Phytophthora species that cause the alder dieback, and is what I'm going to speak now. When I was making these objectives, what I do to myself is making questions. And the first question was like, does the Phytophthora contain viruses? Because these Phytophthora were not described yet in this virus context. So to make this, what I did was a sequencing of all the total RNA that was in my viruses. I extracted the RNA, then I sent it to sequence, and they did the library preparation. I used the Illumina sequencer to obtain the sequences, and then I did my stay in Finland, and I went there to learn how to bioanalyze those sequences with the computer, thing that I didn't know how to start. So we checked the quality with the FastQ, then all these little pieces that I obtained from Illumina were aligned with the Trinity, having big context that then I was able to compare with the BLAST tool from the NCBI against a database that was focused on the RDRPs, that for those who don't know is the RNase-dependent RNase polymerase, that is the protein that is important for the RNA viruses. Without that protein, the virus cannot replicate itself, and then it cannot exist anymore. The results of the sequences were all those numbers, and I'm just showing you this for you to understand that it's pretty incredible, the amount of reads, the amount of information that you can obtain only for extraction. This first column, well, the second column is the total reads, and then after processing and aligning these little pieces that you obtain from the sequencer, you can have this context, this long sequence that you blast and you compare and you analyze to obtain your final results, no matter if you're checking viruses or RNA expression for other things, but it's with this number with what you work. And the question was, does Phytophthora have viruses? Does this species have viruses? And the answer was yes. I was surprised, but finally I did something that for me was just progressing in my thesis. I found that there were 9 new RPs described that I call like this, but these are not official names. I just put this. And the thing was that these new RDPs were bioinformatically confirmed, and they were belonging from 4 different families. That is Buniaviridae, Narnaviridae, iuFOR-UVa, and one family that is not still classified, but let's call it Ormicovirus. The next step was still in Finland. I was just thinking to myself how I'm gonna know when I'm arriving here to Spain which of my isolates have the virus. So with the computer, with Genius, I designed these primers that after that I took the RNA, I transformed it to cDNA, and then I performed the PCR with my specific primers that I previously designed, and then I reveal it into a gel. And what I'm showing you now is what I'm happy these last days, that I confirmed that 3 of the viruses are present in my samples. The primers work, the viruses are present in the isolates, and the next step would be which effects can have these viruses in my Phytophthora, if they are useless, if they are not, let's say. I still have a lot of tasks pending because I said to you that I have 4 families and I only confirmed 1 in these pictures. So I have to do many PCRs, then the genealogy of the viruses, and then the writing, that is the best part of the science, you know. But in conclusion, what I can say to you that is, in total, bioinformatics revealed that I have 4 new families, not new, but new for me. That is Buniaviridae, Narnaviridae, iuFOR-UVa, and the non-classified family that let's call Ormicovirus. And at the moment, what I can say to you that only Buniaviridae, it's in my samples, but the rest of them will be, I'm sure. If you have any other questions about viruses or many other things that I'm pretending to do in my thesis, I'm open to questions, so thank you so much. Thanks for your attention.